Agora está carregando.
Can you see my presentation?
Nós adicionamos quando te apresentarmos.
Ok.
Fica tranquilo, já está aqui prontinho.
Okay.
Acho que já estamos online.
Estamos.
Então, boa tarde, pessoal. Bem-vindos à nossa segunda palestra Magna deste nosso evento online da SBEC, com o tema de morcegos no sul global. Tenho aqui comigo hoje, então, o meu companheiro de missão, o Henrique Hortêncio Filho, e o nosso convidado de hoje, o nosso palestrante de hoje, uma pessoa e um pesquisador que eu admiro muito, o Dani Rojas. O Dani Rojas formou-se em Biologia pela Universidade de Havana, em Cuba, e obteve o seu doutorado em Espanha, na Universidade de Vigo. E na sequência desse doutorado, que foi até quando nós nos conhecemos, o Dani fez o pós-doc dele em Aveiro, na Universidade de Aveiro, em Portugal, em conjunto com a Stony Brook University, nos Estados Unidos. E ele atualmente, depois destas viagens todas, Cuba, Espanha, Portugal, está aqui mais pertinho, é professor do Departamento de Ciências Naturais e Matemática da Pontifícia Universidade Javeriana Cali, aqui na Colômbia. E, enfim, apesar de ele abranger uma quantidade grande de assuntos biológicos com morcegos, a sua pesquisa foca processos de diversificação em escalas macroevolutivas, combinando métodos filogenéticos comparativos com outras ferramentas para entender a diversificação taxonómica e fenotípica de espécies na região neotropical. Muito bem-vindo, Dani, muito obrigada por aceitares o nosso convite. Pessoal, o Dani vai fazer a apresentação dele em inglês, mas ele teve o cuidado de ter algumas coisas escritas em português ou portunhol. Não se inibam de fazer as perguntas em português, em espanhol, em inglês. O Henrique e eu estamos aqui para auxiliar. O Dani é praticamente fluente nas 3 línguas. Então, mais uma vez, bem-vindo, Dani. Muito obrigada. A palavra é tua.
Obrigado. Boa noite. Thank you very much for the invitation. I'm going to try to speak a little bit slow to make this talk to arrive to all of you. And of course, as Maria Joao said, I'm open to any question or comment you want to make at the end of the talk. So before starting to this thing that I call superpower, a new superpower of bats, I want you to meet 2 of the most important beings that I have in my life. And you can see them in this picture. They are reading a book. They are looking at some mammals that became extinct here in South America, but they are not seeing precisely the same pictures. As you know, we humans, we live in a world of colors. We can see like a lot of different colors. All those plants, but with exception, the flower in the middle, the other 2, the orchid and the one to the right, they are from my garden here in Colombia. So these are amazing plants. We can see a lot of colors. And dogs and many other mammals, they also can see colors, but they don't see as many colors as we humans can. So this is how my dog and other mammals will perceive those flowers or anything that could be colored. And if we want to know why we have this contrasting difference in the way we perceive colors, I'm sure many of you already know the answer from your lessons in animal physiology, and it has to do with the type of proteins that we have in our eyes, in our retinas, right? So this is a classic picture of a book of physiology of the human eye, and you can see that we have these specialized cells in the retinas that are called rods and cones, and they allow us to perceive dim light, for example, in the case of rods, or to perceive different colors in the case of cones. But all of these and many other senses that we use, like hearing or smell or taste, all have to do with proteins. Proteins are like the interface that allow animals to know what is happening around. And unlike mammals, reptiles, they are the champions of the competition because they have preserved 4 different type of proteins. These proteins that we call opsins. The ancestor of mammals and reptiles, they probably have had 4 different proteins in their cones, in their retinas, and living reptiles, living birds, they preserve all those 4 proteins. What happened to mammals? Along the way, there were some proteins that were lost from our eyes. And because of that, we call reptiles animals with tetrachromatic vision because they have those 4 proteins. And most mammals, they have dichromatic visions because they only kept 2 of those 4 proteins, right? But what happened with humans? Why I don't see the world in the same way my dog see the world? It's because in our lineage, a group of primates, we have a duplication event, and because of that, we have trichromatic vision, right? So we are like in the middle between most mammals and reptiles. In any case, imagine what, I don't know, a macaw or a hummingbird can see with their eyes is way a more brilliant world, a richer world in terms of colors than the world that humans see. But anyhow, we are still happy with our vision. Now you may ask, what about bats? All this is like a very broad context. Why I'm bringing this to you? Okay, regarding bats, I'm going to say that I'm going to focus on this family of bats, phylostomid bats. And yesterday, you invited Susan Sang, and I don't know if many of you know this, but she is the author of this picture that has been around for a while now on the internet. So she is a great scientist, she's a great conservationist, and she also has a great sense of humor. And it's true when we are talking about bats that phylostomid are like, you know, phylostomids. They are like kind of one of the kind. And this corresponds, of course, to the dietary diversity they have, also to all those morphological and sensory adaptations that evolve in this group of bats. And of course, this is the largest radiation of bats here in the America. And it's the group I've been studying for a long now. And I'm going to focus on some of the senses that at the same time are kind of superpowers of phylostomid bats. As many other bats, phylostomids have a very good hearing. Of course, they use also echolocation. They also can perceive different chemical molecules, either through olfaction, vomeronolfaction, and even taste, of course. And then we know that some phylostomid bats, the ones that specialize in blood, the sanctivore bats, the vampire bats, the actual vampire bats, they can perceive the infrared radiation, right? They have this cool specialization. And then bats also have, at least phylostomids and many others too, have night vision. They have in their eyes, in their retinas, these rod cells that allow them to perceive light at low intensities during night. But in like a couple of years ago, there was this amazing paper that demonstrated that some phylostomid species actually have dichromatic vision. Because it's not enough that you find in your eyes the 2 types of proteins that many mammals, like my dog, have in their eyes. Having those proteins is not enough. We need to be sure that those proteins are doing what they are supposed to do. And in this paper from Alexa Sadier, they found that yes, there are some species of phylostomid bats that can perceive different colors. Not as many colors as humans, but at least they have a dichromatic vision. And this has to do with these 2 proteins that we see here to the right. One of those proteins answer to red and green colors, mostly green. The other protein answer to blue and violet colors, and even ultraviolet. So at this point, I want you to keep this information. Some phylostomid bats can see colors in addition to have very good vision in dim light. Why I'm telling you this? One of the explanations from that paper from Alexa Sadier is that this dichromatic vision would allow these bats to locate fruits. In fact, those bats that have a frugivorous diet are the ones showing the functionality of these proteins. But what if these two proteins are not just for locating fruits? This dichromatic vision maybe can play another role in another function. And because I know you're very clever, based on the title of my talk, you know that I'm talking about this glowing thing that I will explain in a little bit. So this is what we call biofluorescence. It's when an animal have some molecules, some structural molecules that absorb light, and then they can emit a new color of light in a lower energy level. And as you can see, this has been discovered or has been described like in many organisms, in plants, in fungi, in spiders, in scorpions, many insects. And also in vertebrates, there have been some reports, including one from Brazil, if I recall, this little frog that we see here. Sharks, turtles, birds, frogs, they also have this biofluorescent thing. In mammals, one of the oldest reports of biofluorescence was in 1985. I was 5 years old by then, in a group of marsupials, right? Of the delphin marsupials, of possums. And the authors identified like a list of about 12, 15 species, I don't remember. That happened, and I want you to pay attention, please, to the year that appears in this slide. That happened in 1985. Then in January or February of 2019, it was like before the pandemic, but not that far ago, a group of scientists from North America described also biofluorescence in flying squirrels. Because we are all bat lovers, we know that the actual flying mammals are bats, okay? But we know, these gliding squirrels receive this name of flying. 3 species of the genus Glaucomis that occur in North America, they all glow. And you can see here one of the figures of that paper using these skins that they have in collections, where they have this pink color, both in dorsal and ventral surfaces of the animal, showing that they are fluorescing. So when I read that paper, and I remember, I can tell you where I was at the time, I said, okay, so look at this. We have didelphid marsupials, which are nocturnal, crepuscular, which have these 2 proteins that allow them to have dichromatic visions. We have these flying squirrels, which are also nocturnal, which also have these 2 proteins in their crowns. What about bats? What about phyllostomic bats that also have these proteins that are really functional and are nocturnal? Could be that bats show biofluorescence. You already know the answer from the title. The answer is yes. But at that time, first I have to order in Amazon a pair of ultraviolet flashlights in order to check if the fur of these bats actually fluoresce. I was waiting I don't remember how much time, like 7-8 days for the flashlights to arrive. In the meantime, I talked to a colleague here in this town who is a professor from another university, Universidad del Valle. That university has one of the largest mammal collections here in Colombia. So I was talking to him in order to have access to the collection. And as soon as I received the flashlights, I went to that university. I opened the cabinet. I opened one of the first tries. And when I addressed the light beam, everything there was glowing, was fluorescing to ultraviolet. And I was in shock. This was a hunch that became true. So that is not enough in science, as you know. We need to describe that phenomenon. We need to be sure that this is actually biofluorescence. We found that in 77 species of phyllostomid bats from 35 genera, these different levels of biofluorescence, we had to set this very cutting edge, I don't know how to call, tool. You can see the two flashlights. It was the best we have at the time, but this was a pretty good camera to take the pictures. We have to take 8 photos for each bat, 8 for the dorsal surface of the bat, 8 for the ventral surface of the bat. We have to put all that together. We have to standardize the pictures, and then we had to start measuring these values of biofluorescence. You can see this note at the bottom of this slide. There are other families of bats with biofluorescence. I focus in phyllostomid because I'm in love with them, but I dare to say that biofluorescence is widespread, not only in phyllostomid, but also in other families of bats, and I'm sure you're going to be able to describe that if you have the interest. So what happened with this? All this was during 2019, and then even in the field, as soon as we catch any bat in the field, we use the flashlight to check these fluorescence patterns of the fur. Also to be certain that not only fluorescence comes from specimens that have been collected, that are stored in those collections, but also in the real world, in the actual world, in the living bats, they also have that property of their fur. One undergraduate student at that time, he already graduated from biology here from Universidad Javeriana. He decided to do his dissertation on this phenomenon. And he presented the results in the first World Bat Twitter conference back in 2020. That was in May of 2020. We were working on his dissertation. We were trying to test different things that I'm going to talk about later to be certain of this. So at that time, we hadn't any manuscript ready or in publication. But in any case, this was in May of 2020. That same year, in October, you can see that there was this paper of fluorescence in the platypus. And then the next year, in February, they describe fluorescence in this genus of rodents from South Africa. So it was like we opened this kind of worms, and then people were like realizing fluorescence in mammals. And at the same time, we were happy because those early reports on opossums were actually like the starting point to study this phenomenon in the entire group of mammals, and different species are demonstrating that fluorescence in some is a phenomenon that is really, really common. However, there's something regarding all these studies, and is that in some of them, scientists were able to identify in the fur of the mammals, of the different specimens, which molecules were responsible of creating these beautiful colors that you can see here. We were not able to do that because we lack at that time of the what is needed to run this kind of tests. But also there was another question that in any of these studies they were able to answer. They didn't analyze hypotheses related to the ecological role or the adaptive significance of fluorescence in mammals. So we were not able to analyze the fur from a chemical point of view, but because we have this phylogeny and we have so many species, we were able to answer different questions that could help us to understand the ecological role, if there is any ecological role, of biofluorescence, in this case, in phyllostomid bats. So first, you can see here the phylogeny. This is the same tree. It's like a mirror image. Here is the dorsal fur of the same individual. Here's the ventral surface. And you can see here how that trait, how the biofluorescence evolved in this family, the largest circle and the yellow are the one with highest intensity of fluorescence. And you can see that most species are restricted to stenodermatines, to that subfamily, although you can see that some nectar-feeding bats and here some micronectaries, they also show a very intense fluorescence. Then the other question that we want to solve before doing all this analysis about the ecological role of fluorescence is if somehow having bats that were collected at different moments and that were treated to be stored in the collection, if that somehow could have affected the fluorescence of the fur. And in this plot, which is for the dorsal surface of the bat, and this one, which is for the ventral surface, you can see that there is no relationship between the age of the specimen and the intensity of fluorescence, which means that it's okay to work with skins as old as 1980 or 1970 something, and skins of very recent captured bats. They all show more or less the same intensity of fluorescence. Now, with being certain of that, we decided to focus on some hypotheses. And we started with two hypotheses related to interspecific relationships. Then another hypothesis, the third one, about ecological specialization, and the fourth hypothesis about the roosting habits of bats. Regarding interspecific relationship, the first thing we asked was, what if this fluorescence has to do with mate selection? We know that some phylostomid bats, males, they have some display in front of the females, and the female decides if she mates with that male or not. So if they have this dichromatic vision and they have biofluorescence, what if they are using that as a way of selecting a suitable mate? We didn't know, but we wanted to test that. The second hypothesis, also within interspecific relationship, has to do with visual communication. Many phylostomid bats, they form groups of different size, depending on the species. And it has been shown that in mammals, species with very good vision that also form groups, visual communication is important for the cohesion of the group, right? So what if biofluorescence could be used by bats as some mechanism of social communication? The third hypothesis had to do with ecological specialization. The jack of all trades and master of none, if you are, if you, for example, eat like a lot of different things, you don't have enough specializations to be really good at eating one of those different things. Because of that, maybe you don't want to be like very conspicuous. So another bat that is very, very specialized could detect what you are doing and could replace you, right? So in other words, we were hypothesizing that generalist bats will show lower intensity of fluorescence because it's not advantageous for them to be detected than specialist bats. And the fourth hypothesis has to do with the type of roost. Some bats spend the days, in caves, in crevices. Other bats use the foliage, like building tents or just hanging there between the branches. So because they have different exposure to solar radiation, we hypothesize that probably they will show also a difference in the intensity of fluorescence. Those were the four hypotheses, and this is what we found. These are our results. So if biofluorescence plays a role in mating selection, then we would expect that the intensity of biofluorescence would be different between males and females, right? We didn't have the time to mount experiments, to do observations in the field, so we have to do these phylogenetic comparative methods, and we compared the intensity of fluorescence between the two sexes. What did we find? That they were not different at all. Here in purple or violet, I'm very bad at colors. I'm talking about colors and vision, and I'm very bad at naming colors. You can see in this color are the females, the intensity of fluorescence for the females, and in yellow is the intensity of fluorescence for males. And you can see both for the dorsal surface of the bat and for the ventral surface of the bat that they are pretty much the same. So this hypothesis, at least with this analysis and with our data, is not supported. So we moved on to the second hypothesis. So if they use biofluorescence as a way of visual communication, then we would expect that interspecific variation of biofluorescence would be higher than interspecific variation. What did we find? We didn't find that. In fact, we found the opposite. We found that interspecific variation is slightly higher than interspecific variation, which means that these species are not using, apparently, biofluorescence to communicate. And these histograms that you are seeing here is just showing the repetition using different group of species. We were like with 77 species, but because we know that some patterns could be idiosyncratic, we subsampled that, and in every case, we obtained the same result. So it looked like biofluorescence is not important, or is not that important for visual communication in these bats, in phyllostomid bats, or at least in these 77 species of bats. So moving on to the third hypothesis on ecological specialization. We would expect that the broader, the wider the trophic niche, the more different items a species feed on, then the lowest will be its fluorescence. And on the opposite side, the more specialized a phyllostomid bat will be in terms of diet, in this case, it will show like the most glowing pattern of all. What did we find? We find no relationship at all. You can see here niche breadth in the x-axis, and you can see the values of biofluorescence in the y-axis, and you can see that there's no trend, nor for the dorsal fur, nor for the ventral fur. So this hypothesis also rejected based in our data. And of course, imagine that this was the dissertation of my student. So imagine how he felt at this point. Well, we are describing what we think is a pretty cool phenomenon in bats, but we wanted to go a step forward and trying to figure out why they show this fluorescence. And every hypothesis that we were testing, it didn't provide evidence. So we moved to the fourth hypothesis regarding the type of roosting. And we divided bats in those that spend the day in the vegetation and those species that spend the day inside caves, inside crevices. And we didn't find support for this hypothesis either. You can see here the top distribution is the biofluorescence of species that live in the vegetation during the day, that spend the day using the foliage. And in yellow, you have the distribution of biofluorescent values of the species that live without burrows, caves, whatever. And both for the dorsal and ventral fur, you can see there's no difference. But then at that point that we were trying like, okay, we can present this. These are results, right? We didn't find any pattern. This is something that should be known by the academic community. At least the dissertation committee should know this. Then we realized in the phylogeny that some of the species that show the highest intensity of fluorescence are these species that build their tents using some kind of leaves, those tent roosting bats. So we give it a try and we say, okay, let's split phylostomids between those that live in tents and the rest of the species. And here we did find a difference, finally. So we were happy, but we were not that happy because based on this segregation of species, we cannot tell, or at least I didn't feel very comfortable at the time about whether this result actually supports this hypothesis on the type of diurnal frost. So what did we do then to gain more evidence on this result? In this paper from 1985 of the possums, the scientists took samples from the fur and they found that the molecules that make these marsupials glow, fluoresce, are metabolites from this amino acid, from the tryptophan. So this is something, it's some clue, because it turns out that the tryptophan is an essential amino acid for mammals. It means that we mammals need to acquire this from diet, or we need some partners that allowed us to produce enough tryptophan for other functions. Who are those partners? Those partners are bacteria, the bacteria that we have in our gut. So this is a preprint from Melissa and Gala, and I wanted to focus in this part of the plot here to the left. And what we are seeing here is how rich are the metabolic pathways that allow to synthesize this amino acid. And you can see that the fructivore bats that specialize only in fruits, show the most enrichment in their metabolic pathways, meaning that the bacteria they have in their guts are the ones that produce this amino acid in a larger amount, in average, than in insectivore bats or even in sanguivore bats. So this adds to our previous evidence from marsupials, and maybe there might be something there related with what? With roost? No, related with diet. But it turns out that this species of 10 roosting bats are specialized frugivores. And when you're a specialized frugivore, it's very hard for you to acquire proteins from fruit. Because of that, you have all these microbiota of bacteria and other microorganisms in your gut that allows you to produce the tryptophan that you need. And if you have all this large amount of production of tryptophan, more than other species of bats that are not frugivores, and if the biofluorescence of phyllostomid bats depends on tryptophan or on those metabolites that come from tryptophans, then we might be onto something here. You see that there are different if. So it's what we have so far, in part because we haven't been able to check which are the molecules that accumulate in the pelage of bats that make them glow. Regarding that, there's another paper that was very recent, and they analyzed there one possibility of fluorescence, not in bats, but in these flying squirrels. And that possibility is a fifth hypothesis that we didn't test and had to do with avoiding predators. Why? Because some nocturnal raptors, some owls, for example, they show biofluorescence in their wings. So what if biofluorescence is acting as a kind of Batesian mimicry in bats? And there's another possibility, and you can see also lichens showing biofluorescence. So what if biofluorescence in these bats is a mechanism that makes them mask with the background and avoid the predators? We didn't test that because we didn't have data. And in my opinion, this is cool if we test it with experiments, right? So, but it's another possibility. And then there's another paper that relates to this biofluorescence on rodents. These are like the last species of mammals in which biofluorescence was discovered. And in this case, they are not purple, they are not green. You can see that they are kind of reddish. And And also the authors were able to obtain which molecules were causing this fluorescence in these guys. And they found that there are some different kinds of porphyrins. Porphyrins are a byproduct that accumulate in a mammal from the oxidation in the metabolic pathway of the heme groups. The heme groups, the one that we have within the hemoglobin to transport oxygen in the blood. So along the metabolic pathways, to build this important molecule, some compounds are oxidized, get oxidized, and generate these porphyrins, which are bad for mammals, actually. So these authors, Toussaint and colleagues, they propose that mammals have a mechanism to excrete the excess of porphyrins, and mammals do that through the fur. And because of that, they have this glowing pelage. If this is true, then biofluorescence in mammals, it's not related at all with anything ecological. It's just a byproduct of the metabolism of the heme group. But the key point is that this applies only for mammals that show fluorescence in reddish colors. And luckily to us, phyllostomid do not fluoresce in reddish. So the mystery, and this is like the main message I want to share with you today, is that this is pretty cool, in my opinion, to have these fluorescent flying mammals all around. And it's really widespread. But if this is related to any ecological factor, we still don't know. We still have a lot of work to do. And of course, I'm open to any possible collaboration and to your suggestion about all these issues. This is part of my working team. Many of them are undergraduate students. Some of them already graduated this year. This is Matthew from the University of Aveiro. He is doing his PhD. And that's it. Thank you very much for having me. Obrigado for your attention. And I'm ready for your questions and comments.
Dani, muito obrigada pela apresentação. Eu só tenho uma palavra, uau. Acho que é partilhada pelo nosso público. Muito obrigada. Eu vou abrir para perguntas, pessoal. Podem fazer perguntas em português, em espanhol, em inglês. Fiquem à vontade. Enquanto o pessoal se prepara aqui para as perguntas, e tu vai dando aqui os parabéns, eu vou pedir ao Henrique que vá passando aí para tu ires vendo. Posso fazer em português? Se tu não entenderes, diz-me. Dani, tu viste algum padrão mais marcado nas listras faciais ou nas listras dorsais desses stenodermatinae? Porque, coincidentemente, os stenodermatinae são aqueles em que tu encontraste mais padrão, mais biofluorescência, e também são a maioria, não sei se a maioria das espécies, que se abriga em tendas. Existe algum padrão associado a isso?
The problem is that first, we didn't find any pattern. But also in part because some of the specimens were not like very good prepared as to the camera to focus on the faces of the skins, So because of that, we were not able to check that. We did detect something. For example, Ectophila alba and some Platyrrhinus, they show from what we saw, without quantifying, they do show a higher fluorescence in the white pelage in the case of Ectophila, or in the white lines in the case of Platyrrhinus. But we didn't quantify that. But yes, it's certainly a possibility. And maybe, thinking a little bit more on that, maybe it can relate to this hypothesis of social communication, because the stripes certainly play a role in that. So probably if we quantify that difference, if we are able to do that, maybe from lived individuals in the field, something could emerge from there.
Well, I bet a lot of people will now buy a specialized flashlight to go to the field. I was already checking the prices here.
Yeah.
Because Cynthia wrote in Portuguese, I will make the question in English. She thanks you for the talk. She loved it. And while you still didn't find the reason why bats flourish, do you think that that could be an advantage for bats, and why? And if you believe it, why?
Well, that's an interesting question, because I mean, so there's this term from English that I don't find like a very good translation in Spanish or in Portuguese, which is spandrel, or spandrel. It is, I remember, which is like, some trait that evolved and it's just there without playing any role. It's just there. And when my student, when Esteban presented this in the first Twitter conference, World Twitter Conference, one of the people that attended to his talk, he posed that question, and we were thinking about it. So I think that the first thing that we need to figure out is in which conditions these bats will actually glow, right? Because, for example, with this frog that I mentioned in the beginning, it was demonstrated in that paper from the President of the National Academy of Science that the same wavelength in which the frog fluoresces, they have cones in their retinas with sensitivity to that particular wavelength of light, right? So you can see here they're like a strong functional connection. They are fluorescing and they are using that like to locate individuals or whatever. In this case, we don't know. I really can't tell. We were thinking on these four hypotheses, and we were trying to find like a strong evidence, but the evidence we have for the roost hypothesis is kind of weak. We don't know what substances make the fur fluoresce. So it remains an open question, because there's also like a sixth hypothesis that we didn't try, and it has to do with this way of advertising something to other species, like or to other individuals. Like, for example, this is an important trophic resource. We have this tree in blossom, and let's feed on this. And we were not able to test that either. So I think there are many possibilities, and testing those hypotheses, we have to keep in mind that it may have no ecological role at all, and just be a byproduct of the metabolism of bats. That's also a possibility. But it's the ways are open to work on that and see what we can find.
That is the ecologist nightmare. Some trait without any without any function.
Exactly.
So now the next question is from Daniela Bola. She thanks you for the talk, and she asks you if you were able to find, or if you found any bioluminescence in the plants that serve as roosts. For example, the alicornaceae.
We didn't test that. Of course, it's crazy when you are in the field with a group of students and you have the flashlights, you like to to illuminate everything to see what fluoresces and whatnot. But we didn't test that. It's something interesting that we could try, because, for example, for Ectophila alba, many of you probably know, the accumulation of carotenoids in the skin, in the lymph nodes, and in the ears, that yellow color that we have, it has to do with this light, this sunlight that is filtered by the heliconia. So it plays a role there. So we have, I guess, to measure that in the plants that serve as roost, but it's something that certainly deserves like further examination because it could provide some cues.
Well, Adriana's question was really similar to that one. She asked you the same if the plants used for tents would have bioluminescence, or after the leaves were modified, which is also an interesting question. But well, I think that's a question for the future to test.
Yes.
While we don't get any other questions, I have another question, Enrique. I don't know if you have any questions, but I'll ask you now. Danny, let me just ask you. It doesn't have to do with the bioluminescence in bats, but with their ability to see these different wavelengths. So one idea that we have when we read the literature is that there are the bird fruits and the bat fruits. So now this may be a little challenged with this ability of bats to see other wavelengths that we had an idea that they were unable to see in the dark. What do you think about that?
So this is kind of new, all this stuff like new, like in a couple or maybe 3-4 years ago, because something that was game changing in this knowledge on sensory ecology of bats, at least in vision, is that many bats were considered to be colorblind just because the genes were not functional. And then this paper from Alexa Sadier, they quantify not only what was happening to the genes, but also to the messenger RNA, and also the expression of proteins, and also the density of different types of cones and rods also in the retina of these bats. And based on that, we are having like a clearer picture of what's really going on. And I would say that these histochemical tests on the retinas are like the final word on whether the ratio of rods versus cones or the ratio of different cones would actually provide this, at least from the biochemical point of view, would actually provide the ability to see colors and to see dim light. I'm saying from a biochemical point of view, because also having the proteins functional is not enough. We need to have these pathways within the brain to process all that information. And that's also that a neurophysiologist should need to put his or her hands in and figure out. But at least from the biochemical point of view, we are having like this very cool picture of that in phyllostomid, some more moped bats, Noctilio, Lepidinus, if I remember from the paper, they can see pretty well, or at least with the two cone proteins that they have, they can see like kind of a colorful world. Yes, that's it. I don't know if that answered your question.
Totally, and that's completely amazing because that changes our perception of what bats can see in the dark. Because we always say that, especially for echolocating bats, that they basically replace their vision by echolocation, but this can be paradigm changing because now bats may see quite very well in the dark, and I think that is amazing. Henrique, tinhas uma pergunta, né?
Dani, eu tive pensando na luminescência no caso dos morcegos e na possível associação com mimetismo. E daí, no caso, essa coloração teria um aspecto de aposematismo e eles imitariam, então, as aves, né, as aves predadoras noturnas. Mas eles são predados por essas aves. Eles são predados comumente por essas aves. Então, nesse caso, a luminescência ela não seria um ponto positivo para eles, para essa situação. E aí eu queria que você comentasse um pouquinho para a gente sobre as implicações negativas que a luminescência, embora as positivas a gente já tenha ouvido, e muito interessante. Aliás, não te parabenizei, obrigado pela sua fala, maravilhosa. Mas o que você acha, frente ao que você tem lido, estudado, com relação aos pontos negativos da luminescência, inclusive pensando na predação?
Okay, yes. Yes, because we have I'm going to go back to this slide. So we need to remember that birds, like other reptiles, they have tetrachromatic vision. So they are pretty good differentiating colors. They can see many more colors than any other amniote can see. And when I talk to my students, I always say, okay, if we want to know how much are we not seeing, if we compare our vision with that of birds, we need to compare how much a dog is not seeing when we compare their visions with ours, right? So in the case of these nocturnal raptors, they also can detect colors at night. So in that case, as you mentioned, being fluorescent could not be quite advantageous. That hypothesis of the Bayesian mimicry, it was proposed for the flying squirrels in that paper of the flying squirrels. And the author said that maybe if you have an owl trying to hunt something in the forest, and they see one of those flying squirrels gliding nearby, it doesn't attack the squirrel because it kind of confounds the squirrel with another raptor. But, you know, this is like, okay, this is what we could expect in theory. But the other side of the story is that what if that fluorescence is like a beacon saying that, hello, I'm here, come and eat me? Because these birds have the ability to detect the patterns. That's why I was mentioning when I was in this part of the presentation that I think that it should be tested with experiments and see what happened. I don't know, like using some of these markers that are used for ultraviolet signaling and create like some artificial bats and put those in the forest and see how many are attacked by these raptors compared to other artificial bats that won't show would be a material that would filter any fluorescent light. I don't know. It's kind of, we can think like in a lot of stuff to solve this. But yes, one of the negative side would be to be more conspicuous to predators or to competitors, right? Which is something that we try to address with the hypothesis of ecological generalism.
Cynthia asks, is it possible for the babies to glow after they feed on their mother's milk? Do you think that's a possibility?
That would be awesome if we can check that. That would be awesome. I don't know, because, well, if what seems to cause this fluorescence in the pelage of bats is the accumulation of different molecules, and those molecules come from metabolic pathways. So maybe not immediately, but who knows if these baby bats have already their bodies covered with fur, if after lactating they will show some fluorescence that they don't show being when they are fasting, for example. So yeah, that's something that it could be also checked. I don't know. There are so many cool ideas. This is like a whole new world in which we want to jump in and figure out what is really happening. But, you know, time is not enough sometimes.
Well, I think most of the questions are questions for you to test in the future. There is such an amazing world of possibilities to test and to search now, and I think you'll be gathering a lot of data from a lot of collaborators, because I bet everyone here is now just ordering a flashlight to check that. Pessoal, não sei se vocês têm mais perguntas, eu vou fazer uma última pergunta. Olha, a Adriana escreveu, it's in Spanish, so because you mentioned noctilio and pteronauts, do you think they would be able to understand the fluorescence or the iridescence of the scales, for example, from fishes or from lepidopterans?
I don't know, but that's also a cool possibility. Yeah, it would be awesome. I don't know if nocturnal butterflies show biofluorescence. But yes, it's yeah, you have so many great ideas, and I was like, okay, I want to devote the rest of my life to solve all these questions because they're really cool. But I don't know. I think, I mean, maybe, and it's like the common signal that you can take from all those papers talking about fluorescence in mammals is that nobody has shown yet why this is actually happening. So we don't know. Maybe there will be some paper in the future that will demonstrate that these other colors of fluorescence are also a metabolic byproduct, like it happens with the reddish fluorescence. Or maybe someone actually find like an ecological explanation. We don't know. It's, you are invited. I can talk to you about what we did, what software we use, what analysis we perform. You have my email there if you want to write it down and to discuss about any idea you may have if you want to study biofluorescence in bats or in any other mammals. Just to deliver some information, we had here in the collection I visited here in the other university in this town, they have a lemur, a ring-tailed lemur, like the one from Sovumafu. I don't know if in Brazil you watched that program, probably. They show fluorescence. So fluorescence might be also in primates, and someone should have to deal with that somehow. And we found fluorescence in other orders of mammals in addition to primates and bats. But that information I cannot tell you right now because there's someone trying to work with that. But it's amazing. I mean, maybe being that widespread, either you have like the same ecological significance, which is kind of hard in so many different groups, or it might be the possibility that in different group of mammals, the phenomenon evolve answering to different pressures, or it might be just that it's there because of what happened with the fur of mammals. Who knows? Let's see if we find out the final explanation of biofluorescence in bats and in mammals.
Well, Danny, I think that wraps our talk very well. Muito obrigada a todos que estiveram connosco hoje, obrigada ao Dani por ter generosamente partilhado o seu tempo, o seu trabalho, estas descobertas incríveis connosco nesta semana de SBEC Online, ao Henrique por estar aqui comigo, fazendo companhia e auxiliando aqui nas partes mais, enfim, tecnológicas da coisa. E convido vocês, né, para continuarem a assistir às nossas atividades ao longo do resto da semana. Amanhã temos então os nossos póster, vídeo, também aqui no YouTube. Então venham assistir, venham deixar os comentários e as perguntas de vocês para os nossos palestrantes. Revisitem a nossa conferência de morcegos brasileiros no Twitter, trabalhos muito legais, com temáticas muito diversificadas. Na sexta-feira temos então o encerramento do nosso evento com uma mesa redonda sobre conservação, energias renováveis, mineração e mais uma palestra magna com a Mónica Dias, do Programa de Conservação de Morcegos na América Latina, que acho que vai encerrar muito bem o nosso evento. Embora nós tivéssemos um evento que era Morcegos no Sul Global, infelizmente nós não conseguimos ter um palestrante de África. Estava programado, estava confirmado, mas o nosso convidado acabou por ter uma urgência, seria a nossa palestra de amanhã. Mas, e em cima da hora não conseguimos um substituto ou uma substituta. Fica prometido que os morcegos africanos serão em breve nos nossos eventos representados. Então é uma falha nossa no nosso evento, estava tudo bem planejado para, enfim, percorrermos aqui o Hemisfério Sul, mas, enfim, estes imprevistos acontecem, mas que certamente numa próxima oportunidade teremos esse palestrante connosco aqui online, ou quem sabe presencialmente num evento da SBEC. Pessoal, boa noite a todos. Henrique, Dani, adorei rever-te, muitas saudades, foi um prazer ter-te aqui connosco. E até uma próxima.
Valeu, pessoal.
Obrigado.
Boa noite. Dani, muitíssimo obrigado. E que bom que essas palestras vão ficar disponíveis no site da SBEC, no YouTube da SBEC, pessoal. Passem para os colegas, né? São temas imperdíveis, com falas imperdíveis, com pessoas maravilhosas que têm muita coisa para ajudar a gente a construir nosso conhecimento. Valeu mesmo e a gente se vê. Um abraço.
Obrigado.